So next up, we have a very exciting speaker, Dr. Cardon. He is an internationally recognized human geneticist and a demonstrated leader in academic, pharma, and biotech research. He joined the Jackson Laboratory as President and Chief Executive Officer in November 2021. Previously, he was Chief Scientific Officer and Chief Scientific Strategy Officer at BioMarin Pharmaceutical, a rare disease biotechnology company. Before his role at BioMarin, Cardon was Senior Vice President and Head of Genetics, Quantitative Sciences, and Alternative Discovery Development at GlaxoSmithKline. He also spent the first half of his career as a senior academic. He was a faculty member at the University of Oxford, initially as head of bioinformatics and statistical genetics at the Wellcome Trust Center for Human Genetics, and later as a Wellcome Trust Principal Fellow and Professor of Bioinformatics. Prior to that, he was a professor of biostatistics at the University of Washington and co-chair of the Herbold Bioinformatics program at the Fred Hutchinson Cancer Research Center in Seattle. He has authored more than 225 scientific publications and 15 books and chapters, mainly focused on genetics, methodology, applications, and discoveries in rare and common diseases. He is an elected fellow of the United Kingdom's Academy of Medical Sciences and the American Association for the Advancement of Science. And he has an amazing announcement to make. Thank you, Lon.
Note to self, I need to write a shorter bio. Thank you so much. That was terrific. I really appreciate that. It's a great pleasure to be here today, and I do have an announcement to make for you. Let me, before I start my own presentation, give a plug for the previous one with Megan. Rare diseases are so important to the patient community, but to the overall trajectory of science and biomedicine and new drugs today. And even though they're rare, they're not tackled individually. So I just want to get right behind exactly that message that you were giving. I do have an announcement to make for you today, and I'm hoping that I can build up an understanding of why we see this as so important and not just yet another acquisition or another merger, because our view is we have the potential here with something that I'm going to announce to have an impact on much greater of a community than just the organizations and in fact in all of drug discovery and all of biomedicine throughout. So I'm going to try to build this up for you a little bit, the rationale, and then I'll go into the partnership or the acquisition itself. So, it's easy to forget right now in these trying times we have with funding and support and science that this is the best time in biomedicine we've ever had. It is amazing right now. This is a picture of someone that the community calls Baby KJ. You can read on the side what happened, but the story goes like this. Baby KJ was, as so many are, diagnosed with a rare disease very early on in his life, and it did not look good and was probably terminal. And it's one of those situations where you go to the physician or the counselor and you say, what are my options as a parent? What can I do for this with this terrible news? And the response is usually spend as much time and give as much love as you possibly can, because that's all you got. That was the situation here. And in 6 months, a drug was made specifically for Baby KJ. Put in Baby KJ, and Baby KJ is thriving. 6 months. That doesn't happen. Drug discovery takes 15 years and billions of dollars. And here you got 6 months. You got to run. So I start with this because this isn't hyperbole anymore. This isn't something that we can go strive for. We have this today, the availability of the technologies and the science and the collaborations that if we really want to do it and we can put our minds together, it can happen. And it just did. So it's an amazing story in science. And whenever you get negative about funding or whatever we are, remember, this is what's possible. And we want to expand on that. So how do we scale something like this? How do we get this so that we can treat all the baby KJs out there and all the diseases that actually, in rare diseases, afflict 1 in 10 of us? So they're individually very rare, but they're collectively very, very common. So how do we do that? So that's the motivation. Now I want to step back and give you the problem that we're trying to solve. So I come out of, as Nancy kindly said, half of my life as a basic scientist, academic researcher, and half of my life as a drug discoverer on the industry side of things. And so I know we have a diverse audience, so my cartoon for you is to describe drug discovery in a really simple term, okay, in a really simple framework here. On the right-hand side there, in the word cloud, you have clinical work, the clinical trials, right? You've got a medicine, an investigational medicine, you're going to put it in humans, and you're going to ask, does it do what I want it to do? Is it safe? Does it do something that's useful? That's where all the money goes, and that's where you see the big failures. I'll give you an example. I worked on a cardiovascular outcome trial, one trial, and those type of trials are particularly challenging because you need tens of thousands of people because you literally just watch and wait for the outcomes, in this case heart attacks, to occur. And so you have to watch lots and lots of people to get enough events. $600 million on one study. So these aren't made up numbers. These are real numbers. And it so happens that one failed. So that happens, and the result of that is all the bad things that we like to complain about as citizens, that drugs cost a lot of money, we want to poke the finger at big pharma, that they're greedy or whatever, but actually it costs a lot of money because you get all that failure on the clinical trial side. And there's not a whole lot you can do to improve that side of the equation because you're still going to want to test new drugs in humans before you distribute them out to the policy. So you kind of got to follow the cookbook on the clinical trial side. On the left-hand side there is basic research. Everything. We've tried to just kind of capture. There's a word cloud of a whole bunch of stuff. Think genetics and proteomics and biology and physics and everything that helps us understand how humans are wired. And that's working beautifully. That over the past 20 years has been nothing short of phenomenal. We have gene editing with CRISPR, we have the human genome, we have stem cells, we have all these things that have come forward and all these discoveries. We know tens of thousands of genes that influence common complex diseases. Tens of thousands. At the turn of the century, we had 10. We had Alzheimer's disease, ApoE and Alzheimer's, we had breast cancer, BRCA1 and 2, we had a couple. That was it. So we got this kind of a strange paradox. On the left-hand side, basic science has never been better. It's awesome. It's just pumping out really, really interesting things. On the right-hand side, we're still failing like crazy, and it costs more and more and more. We got a problem. We got a disconnect there between these two. And somehow, what we've got to do is use that left-hand side of basic science to better inform the right-hand side so the failures decrease and the timing gets shorter. That's the problem. And that's not our problem. That's our problem. That's the industry's problem, that's the community's problem, it's society's problem if we want to tackle new medicines at a time and scale that we can afford them and get them through. So that's the clinical trial side. I kind of jumped to the punchline. Every few years, I haven't seen the most recent one, this one's a really old quote, every couple of years Tufts University puts out a study on how much it costs to get a drug to market if you count for all the attrition. This was like 2014, it was $2.5 billion. And so now, I don't know what the latest numbers are, but they're going to be $4 billion, and that's why we're paying what we're paying, because that's the situation. On the left-hand side, if we could somehow bridge from all that basic science which is going so well, and better inform the right-hand side, so our best opportunity is to figure out how to turn those discoveries into shortening and increasing the probability of success. That's it, right? So you got science on one side, you got translation of that science on the other side, and we need to bridge it. And if we can bridge that better, then we win. So that bridge, historically, is done in a really simple way. You go through, you do all your basic science, the whole community comes together, they publish papers for years, and so you referred to it quite rightly. The in-between part of those two is called the Valley of Death. Industry's called it that for a long time because you got basic science on one side. and translation on the other side. So what happens there is you go do all the science, hopefully you come up with some sort of molecule or some sort of treatment that's investigational, and you begin to test it before you put it in humans, because we can't put these things in us until we have a decent sense of what they're actually doing and are they safe and effective and that sort of thing. And traditionally, that's done in animals. All right, so you do, and this is how it works today. You do an animal study and you say, okay, did it harm the animals? Is it showing the type of attributes that we want to go forward? That's how it's done. But there are actually 3 ways to do that. There are animal models. I'm going to tell you about the Jackson Lab in a moment. The Jackson Lab is the inventor of the use of laboratory mice to study humans. 100 years old, 96 years old, will be 100 pretty soon. And for the very first group ever, sat on an island in Maine to study tumors and hope to inform something about human cancers at that time, well before the genome era. But that's not the only way we could develop a model before we put it into humans. There are 2 other ways. I only see 3, actually. I don't think there exist any other ways to take a drug forward than 3 ways. You can stick it in an animal. You can put it in a dish, which I'm going to tell you about in a second, with cells. Or everybody's favorite right now, and I'm sure I'll offend somebody in here, we can just magically wave a wand and AI is going to solve it for us. And there's a lot of money going into that AI side of things right now. I think there's probably a place for it. It's early days, and it's in the hype cycle for this type of activity because the reality is we just don't really know how the human body works, and so how are we going to train it? And so it's kind of hard to do that. But those are the 3. You're either going to do it in a computer, you're going to do it in an animal, or you're going to do it in a dish. And the dish side is really, really interesting, which I'll talk about in a moment, and it's part of the deal that we've come forward with. So these are the 3 approaches, and just to belabor the point, so we're the mouse people, Jackson Lab. We're going to tell you about us in just a moment. These are great, and I like to say, and you can fact check me because I've accepted that I'm probably wrong on this, but because the Jackson Laboratory invented the use of mice for biomedical research, others use them now, but virtually all laboratory mice that are used have derived somehow from that Jackson Lab. So if you take the extension of that, every drug ever approved in man has gone through the Jackson Lab or had a hand in the Jackson, every single one of them. So that's a pretty impressive little track record there to have. The part that is great about a mouse or other model organisms that are living is they have all the organs that we're interested in. So we build a drug because we want to do something for the heart, and we can see if it has damage with the liver, right? And you can't always get that with other systems, and AI may or may not be able to tackle that. But in an animal, in a whole animal, it's all there. So you get the off-target effects, and that's really impressive. But what it's not is a furry little human. It's an animal. The gene correspondence between animals, between mice and humans is remarkably similar, but they're not 100%. They're not furry little humans, and so sometimes they will not reflect what's going on in the complexity of a human. Or sometimes investigators will use, we have thousands, and the Jackson Lab has 14,000 different strains of mice. So whatever problem you want to work on, we got one. We got a mouse for that. But sometimes as investigators, we use the wrong ones. And then we blame it on the mouse. We're sort of the craftsman blaming his tools on that one. But for whatever the case, they're a model, and they're an imperfect model. If you go to cells, which I'm going to show you a little picture of in a second. So in 2012, a Nobel Prize was awarded for something I still find. I'm not a stem cell biologist. You're going to hear from one here, a couple of them here in just a few moments, so you can ask them your questions. I see this nothing short of science fiction and magic. You can today, and for this procedure, it was awarded the Nobel Prize in 2012. You take a piece of your skin. Your skin has all your DNA, all of it. It's in there. It's your piece of skin. You can convert that piece of skin into sort of a primordial state, and then forward convert it into a little beating heart, or a piece of liver, or a neuron, or whatever. Your skin, 100% your DNA. If you're a patient, if you've got pancreatitis and you're trying to go for something, it's that DNA. You don't know where the magic and the secrets are necessarily in that genome, but wherever those secrets are, they're in that dish. That's pretty impressive. And you can build the organ or a mini version of those organs to study. Fantastic. What science that is. So that's an alternative. The downside of those, at least today, is you don't have the ability to study the heart and find out what it's doing in the liver or else. So you got a kind of a complement here going on, right? And then we've got the AI side, and I'm not trying to be as pejorative as AI as I sound. We've been here before, and the hype cycle comes, and I do think it will do magic things today, even in science and in industry. It's doing amazing things for problems we already know how to solve. It can do them far better and far faster. But for problems like human biology, we haven't really. It's not going to substitute for all the things that we have done, at least not right away. So the three of those are sort of the golden triad. And so here's the stem cell. I don't know if this works or not, but if it'll work here. On the bottom right there, if I could figure out, this is a user error, I'm sure. If I could figure out how to do it, you can hear from the next speakers on how this process is done. But on that bottom right, a thing that kind of looks like a barbell, if I could make the movie, sits there and beats from a piece of skin. It's pretty cool. It's really, really interesting to be able to do that. So the reason this is so important from the drug discovery perspective, so I had the good fortune or the challenge in my big pharma days. I was a decade at Glaxo, and I lived and ran the interface of that valley of death. So you take the basic science, the discoveries, you synthesize it all down, you've got the drugs, and you're just about to write a really big check. And I had to chair the committees and all those that made that decision where to go forward. And so, I don't know, I got to see lots and lots of projects over the years, hundreds probably. Never, ever did I see a project that was so obvious that the decision made itself. That said, oh, we put it into this mouse, 100%, we know that thing's going to work. Never. It's science, we don't understand. And so we would make these decisions with incomplete information. And as the result, here we go back to, we have all those failures where it costs the most. And it's not a blame of the scientists. The scientists are doing their very best. It's not necessarily a blame of the animals they used or the studies they did. You just didn't have the information. So imagine a world where you could put it together, where you're complementing all these things. You've got the human data to rely on. Okay, that's telling me it's doing what it wants. Then you stick it in a mouse maybe, and okay, well, it's not harming other organs. That's pretty good. And maybe you link this together to try and figure it all out by AI when it emerges. That's bringing the three. That's the dream. And if we could do that, what happens? Well, we get societal impact. The cost of the drug failures goes down. The speed goes up. We get more pancreatitis things through. We get baby KJ coming around. Things change, and they change in a big way. So that may sound like a bit of hyperbole, but it's actually not. That's where we are. There's no aha moment here. All these pieces exist, every one of them. We've got them, but we haven't put them together. So, this is my version of AI, just to show you. So what we want and what we argue, and I'll get to this deal that I'm about to describe. What we argue, and we're one of very few that argue this, it's not that we just need the dish and stem cells. It's not, as we could say, hey, we're just going to stand back and use animals. We're the mouse people. That's going to get you there. And it's not AI either. It's all of them. We need a seamless system. Imagine that world where the cells inform your mouse and the mouse informs the cells and the AI says, none of that makes sense. That's what we need, is all three of those. Now, the timing on this is quite interesting because we just got lucky. We set up our strategy to try to bring these things together now nearly 5 years ago. And the community, NIH, FDA, has kind of woken up to the fact that, hey, these other models could be really informative. Now they're going for the hype and they're going for the big sensationalist sell and say we don't need animals anymore and we don't need this. That's kind of ridiculous because if I were still in drug discovery, I would never put an investigational drug in a human, having not tested it in a full. That's dangerous stuff to do that in this day and age. But the reality is, hey, let's use these other models. Let's bring them all up to the level so they're all valuable. So actually, this is the future, and we see that, the combination of these as the future. And I'm going to tell you about the Jackson Lab now, but the deal I want to tell you about is there exist, I'll tell you about the Jackson Lab as this mouse organization, the founders of the use of that. There exist others that use mice, and that sell mice, and that produce mice, and all that. There exist many others than the New York Stem Cell Foundation that work on those little mini beating hearts and how to derive them. There exist plenty of others in the AI world that we all know about, and they're Googles and Microsofts and so on. There is none, not one, that brings them together. Zero. Not one. And that's what we need. We don't need the independent ones. Otherwise, I'm still going to be sitting in big pharma saying, okay, the mouse gave me partial information, and then the cells gave me a little bit something else. I'm still going to be throwing darts at a really big check. So to the extent we can bring them together, that's what we need to do, and that's the deal I'm about to let you know about. So you can read the slide, but I gave some of it away to you. Jackson Lab, this lovely organization. We're now in, we have 13 sites across the world, but started out with little blueberry crates in Maine, charmingly so, using mice and selling them for 10 cents or something in blueberry boxes. Now we've got 3,000 people across all these places. We've got a holding pen in Heathrow, and we've got sites in Japan, and something like 2,400 institutions across the world use, basically all the biomedical research institutes across the world use the Jackson Lab mice, 14,000 different genetic strains. We've got this big repository. So we're the mice people. Here's where you go, and this is where we distribute them all over the place. But we don't have a deep history in the others, in the AI and the cells. Somebody needs to bring those pieces together. What you've got here in New York, well, this is what I'm announcing, but what you've got here in New York is the New York Stem Cell Foundation. Not 96 years old, but 20 years old, built to take forward, if you remember, initial stem cells were derived from embryos, and that had a lot of questions and societal implications and so on. And those are no longer necessary in what this group has done, and funded and brought together throughout New York to make it a real powerhouse, the ability to do that same work and differentiate those cells without the use of embryos. And what we have announced, and it's the timing of this meeting couldn't be better, we announced we agreed terms for the Jackson Lab to purchase the New York Stem Cell Foundation. We agreed those a couple of months ago. The process is for nonprofits to go through the Attorney General's office. The Attorney General approved on Wednesday, and we completed on Friday. Just Friday. So, and we announced on Monday. So I'll give, Now, I'll just tee this up in closing. So the New York Stem Cell, and the architect of this right here is in this audience. You're going to hear from him in a panel in just a moment. As I said, there are others that do this type of work, but what they've done really, really well is automate a lot of that and build a quality system that we can. One of the things we can do in mice is we have such high quality standards. If one of you in this audience bought Jackson Lab mice, and then I came in 10 years later and bought the same mouse, it's the same mouse. It's the same, it's like identical. So for science, that reproducibility is essential, because otherwise we get different answers. We have no idea why we get different answers. That's what they are building for cells. Set the standard for all cells so that quality is the same lab to lab to lab, generation to generation to generation. And the view is we put that together with the standards and the throughput that we have with the mouse, and there is no other. And we change the overall paradigm. Hopefully everybody jumps into it and somebody else tells us they invented it, fine, because we move the needle forward and drug discovery goes faster and gets cheaper. So I'll stop there, and I think I will. Not very adept at this, or maybe that's the end. We'll leave it there. And I'm happy to take questions. Thank you very much. Well, what you really want to do is hear from the panel anyway, because I think, so these are real scientists at the bench. Please do ask your questions about how this stuff, some of it's really magic, but please, Nancy.
What do you estimate will be the reduction in drug costs with a process like this?
Yeah. Well,
And time
yeah,
of development.
the time is going to take time to implement because we've got to get the community to realize that it's better to use all the tools together. Right now, what you're happening, it's typical, it's capitalism. You know, the people are trying to raise money that are in AI. And so the pitch they want to give is, AI is just going to take over. We don't need the rest of you. That's not the right pitch. Actually, the right pitch is we need everybody. It takes a village. It takes a big village to do that. So when they get past that, then we're off to the races. But if you could imagine, we did some calculations on this a few years ago, but let's say we use that $2.6 billion number, and that's factoring in 90% failure. Just move the needle 10%, and look at what we don't have to crack the whole biomedical problem. Just move it a tiny bit, and we save billions and years in a process that's desperate to need it. Because if we don't do that, there's no reason to believe the trajectory is going to change. We're doing the same old thing we did 5 years ago and 10 years ago. So we're standing to save billions of dollars off the price and the real cost of making innovative new medicines for society across the board. It's a real impact.
I think that's a pretty spectacular announcement, don't you?